So good morning everyone. Once again, I, Dr. Ravneet, on behalf of Organizing Committee BDE, extend a warm welcome to one and all present here in this virtual platform of 2 days online workshop on bioinformatics and drug engineering, organized by Amity Institute of Biotechnology, Amity University, Rajasthan, in association with IEEE Rajasthan subsection. We are grateful to all our participants for accepting our invitation to be a part of this intellectual workshop. Over the last few years, computer-aided drug engineering has become a powerful technique because of its utility in various stages of drug discovery, employing many advanced features. Also, the recent outbreak of this deadly COVID-19 virus pandemic posed serious health concerns around the globe. The lack of approved drugs or vaccines continues to be a challenge and further necessitates the discovery of new therapeutic molecules. Drug engineering has helped to expedite the drug discovery and development process by minimizing the cost and time. We hope this workshop will bring new insights for our participants, amalgamating bioinformatics and drug engineering. So moving on further, I would like to invite our first guest speaker of the day, Dr. Dinesh Gupta. Dr. Dinesh Gupta completed his PhD from All India Institute of Medical Sciences, New Delhi. For his doctoral work, he worked on developing computational biology techniques to study membrane-ligand interactions. His thesis yielded novel insights into the interaction of different ligands, including drug molecules, with membrane phospholipids. He joined ICGEB in 1998 to develop its computational biology program. He also received World Health Organization funding to establish a regional bioinformatics research and training center for Asian scientists working in tropical diseases. He was awarded a prestigious WHO capacity building grant to research at the University of Pennsylvania and Wellcome Sanger Institute, which also helped him to contribute to the PlasmoDB, a web-based resource for malaria researchers. He serves on various editorial boards and is also an active reviewer for several computational biology journals. He is a member of the Scientific and Technical Appraisal and Advisory Group of the Department of Biotechnology, DBT, Government of India. He also serves as a member of the DBT's Technical Experts Committee on Theoretical and Computational Biology, Bioinformatics, AI, and Big Data. Currently, he is serving as the group leader Translational Bioinformatics Group at the International Center for Genetic Engineering and Biotechnology, ICGEB, a renowned institute in New Delhi, India. He has also published more than 100 articles in peer-reviewed journals. His current research interests include computational biology and artificial intelligence in solving problems in healthcare and medicine. For example, recently his group has conducted different projects to study the important drug targets in malaria as well as COVID-19 to design inhibitors against the targets. In addition, his research group has recently developed an AI-based method for quantitative prediction of the anti-SARS-CoV-2 activity of the molecules. His group also developed a deep learning model to classify patients' test X-ray images into COVID-19 and other lung infections, which is the need of the hour. In another application, his group has developed a machine learning model for prognostics of hospital-admitted patients. Currently, he is collaborating with several clinical groups to work on different challenging classification problems in ophthalmology, cancer, and radiology. With these few words, I would now hand over the session to Dr. Dinesh Gupta. Sir, can you please continue?
Thank you, Ravneet, and good morning to all of you. And before I start my talk, I would like to thank Ravi and Manishita for inviting me to deliver a talk on this workshop. And a very good introduction has already been given by Professor Bhatnagar in the inaugural session, and I'm sure there is no need to repeat what he has already talked in his inaugural address. So I will straight away begin my talk, which is on bioinformatics for COVID-19. So these are some of the research interests of my group at ICGEB, which has been already enumerated by Ravneet. So I will not going to read them. So bioinformatics has played a very important role in managing and doing research during the pandemic since its onset in December 2019. There have been many applications from bioinformatics which have been very useful for research as well as managing the pandemic by various stakeholders. For example, bioinformatics helped in managing the real-time data. For example, epidemiological data, example, spatio-temporal disease spread. It also helped in studying the mutations of the virus as it was spreading. It helped in performing phylogenetic analysis. What are the strains, how they are different from the previous strains, and how infectious are these, and taking a stock of all these things along with their spread across the globe. All these things have been facilitated by bioinformaticians only, along with the IT guys. So bioinformatics has been tremendously useful in studying the disease transmission and modeling what could be the future transmission and infectivity of various strains. All these have been modeled by various bioinformaticians, and many of these studies have been very useful in managing the disease and doing research to mitigate the spread of the virus, as well as bring out new therapeutic interventions to control the viral infections. And as far as reference data is concerned, so there are lots of data which were generated by various groups. So they were put into various databases, like databases on protein-protein interaction. For example, the proteins of host and viruses, how they interact and how they are able to initiate infection and viral growth, and how the proteins are interacting once the infection is set in the host, how the proteins are interacting, how they are bringing about the various presentations of the disease when a person is infected. Along with protein-protein interaction data, lot of genomics data was collected. So that is also being managed by various bioinformaticians by developing various useful databases, which are, most of them are real-time, and they are developing as the disease is spreading. So a lot of reference data is also available for drug development. For example, repurposing of drugs which are already approved, since it was an emergency to bring out drugs in the market. So various groups across the globe, including many experimental scientists and bioinformaticians, tried various methods to explore the drugs which are already approved by FDA or other drug controlling authorities across the globe, to see whether these drugs can be used to cure COVID-19 and its effect or not. A lot of data was also collected for various image based platforms like X-ray and CT scans, which were also very useful, and they are also used by various groups to perform different research activities. A lot of data was also submitted to help research which is being conducted to predict the disease outcome, as I told you earlier. So different fields of biology and biotechnology were immensely helped by various kinds of diverse data collected and managed by various bioinformatics and IT groups during this pandemic. So here is a very good table which gives a list of very useful tools that were quickly developed by various research groups. Most of them were already working in various research centers focusing research on bioinformatics. For example, for epidemiology, there were groups like JHU or John Hopkins University website, which gives a real-time spread of the virus in various countries across the globe, which was very much used by various stakeholders who are managing the disease. WHO dashboard is another useful website where World Health Organization is maintaining the information related to the disease, its spread, and vaccination, etc. Similarly, all these websites are giving a very good information about various aspects of the virus and the pandemic. So I will not read out all of them, but here is the reference which you can follow, and you can visit these sites to get information about various aspects of the virus and the pandemic. Particularly, there are few resources which I would like to highlight. For example, for virus genome, are very good resources like GIS-8, which is a global initiative on sharing all influenza data. And this initiative was already in place. The website was there. The database was already collecting data related to various influenza strains. It was quickly upscaled to collect data about the SARS-CoV-2 virus also. And luckily, various countries joined hand in submitting their genomic data to this resource, which have been very useful. Apart from that, NCBI also developed a resource which was already in place. It is not new. And they also opened a separate database for SARS-CoV-2, which is also being used by various scientists across the globe. So this is the website where you can see a snapshot of NCBI virus. You can find the data based on various parameters like searching by a sequence. If you have a sequence of a new strain of the virus and you want to compare it with other strains, that you can do, or you want to search a virus according to their name or strain and so on, that you can do using this resource. So this is a snapshot of GIS-8 website where you can see that 75 lakhs 15,480 genomic sequences have been submitted till today, and it has been very useful in providing information like real-time spread of various strains across the globe, and which has tremendously helped in various modeling efforts by scientists across the globe. And it is also helping in people submitting their sequence and getting it genotyped to know which of the strain is prevalent in the sequences which they are submitting. So this has also helped tremendously monitoring the spread of various strains across the globe, helping the governments to take proper steps to control the spread of pandemic in their countries. So this is another resource where COVID-19 or SARS-CoV-2 related data is submitted related to various chemical compounds which are being used in various clinical trials. So specific resources are being managed by various stakeholders and scientists and research labs across the globe to provide very good utilities related to different aspects of the virus research and development of drugs or vaccines. So here is another website, which is COVID-19 disease map, which is maintained at University of Luxembourg, which also provides very good information about various disease map related to the SARS-CoV-2 virus host interactions. So it is a collection of various high throughput studies which have studied the interaction of various viral proteins and host proteins in details. So these can be used for developing new therapeutics against the virus. So this is a snapshot of Johns Hopkins University's website to show the real-time spread of the virus across the globe. And you can see various countries, and based on the proportion of spread in each geographical location, the circles are drawn proportionately. How many deaths have been reported and how many vaccines have been administered in real time is being monitored very nicely on this dashboard developed by Johns Hopkins University. Indian government also woke up to the uh, pandemic's requirements, and they formed a consortium of various uh, genomic centers across the country, and they formed a consortium called INSACOG. And each one of the participating institution was given the responsibility of sequencing various SARS-CoV-2 samples and submitting the genomic sequence of the sequences collected by them. So eventually these were also collated and submitted to GIS-aid website to share the data transparently. So this has been a very good initiative of Government of India to manage the pandemic. And Department of Biotechnology also started a very ambitious project during the pandemic, which is called as IBDC, or Indian Biological Data Center, where the aim is to develop a very good large database of sequences which are specifically from India. So this will not only collect sequences of viruses which are found in India, it will have genomic sequences from plants, it will have proteomic sequences from plants, animals, and metagenomic data, proteomics data, sequencing data, everything, image, including clinical trials, image analysis, and also and so on. So the idea was to have a platform where Indian scientists can submit their sequence, archive it, and also retrieve data sets which are specific to Indian scenario, which they can be used in their research. So this is a very ambitious project. And to start with, they have already opened a portal to collect information related to the SARS-CoV-2 genomic sequences. So INSACOG partners are expected to submit their sequences through this portal. And IBDC has already tied up with various international centers so that whatever sequence you are submitting to the Indian website is transparently and automatically submitted to the international databases. So you don't have to submit a sequence which is related to Indian strain or genome, and simultaneously submitted it elsewhere. So you will be given a unique ID right here at the Indian website, which you did not have to submit separately on the international website. So this is just to show that the virus is single-stranded RNA virus, which is around 30 kb, one of the largest RNA viruses, and it codes for around 29 proteins. And many of the proteins are being explored as drug targets because of their very important function for the virus in infection and growth of the virus. So today I'll be talking about our research on the spike protein and the main protease of the virus, which is helping in its growth by processing of various proteins so that the virus is able to grow inside the host cell. So before I start talking about my own research, is just to show that several of my colleagues at ICGB are already working on different aspects of SARS-CoV-2 research. For example, Dr. Khanna is working on phytopharmaceutical drug candidate against the SARS-CoV-2 virus. And another colleague is working on development of monoclonal antibodies. My group is mainly focusing on bioinformatics aspects. We worked on sequences as well as chest X-ray images, how to classify chest X-rays into uh, belonging to COVID-19 patient or non-COVID-19 patient and so on. One of the other group is also working on developing novel COVID-19 diagnostics. And ICGB is also working as a diagnostic lab for the neighboring hospitals. So this is just to show that several proteins of the virus are being explored as drug targets to develop novel therapeutics against the virus and the disease. Many of them are targeting different proteins. For example, they are even targeting the host receptor ACE2, or they are targeting spike protein of the virus itself. Some of them are acting at various other important proteins of the virus like RDRP, so and 3CL-Pro, which is a very important protease of the virus, which is very important for virus to grow, and so on. So all these are various stages of the virus infection, entry into the host cell, and its reproduction within the host cell. So different drugs are believed to act on different proteins, and many are being already in clinical trials, and efforts are still on to develop some more drugs. So so this is just to show that in one of the work that we have done during the pandemic was to study the effect of mutations on the spike protein. So we studied the various mutations, how they are affecting the stability of the spike protein, because spike protein is the very important protein which provides anchoring to the host receptor at ACE2, and it helps in internalization of the virus after its interaction with ACE2 receptor. So it has been observed that this spike protein is amenable to many mutations as the pandemic is progressing. So virus is becoming more and more infectious. And what are the mutations and how it is affecting the stability of the spike protein, that it is enabling better infection or better interaction with the host receptor. That was the aim of the study. So the spike proteins or spike glycoproteins are the viral structural proteins that facilitate SARS-CoV-2 host entry. The work was focused on mutations in two functional domains, especially namely which are receptor binding domain RBD and heptad repeat HR1 domain, that are majorly responsible for stability of the SARS-CoV-2 spike glycoprotein, and they play a key role in host cell attachment and infection, as I told you earlier. So these are the mutations that we studied on RBD, and these are the mutations that we studied on HR1. So I will skip few details here so that you get the main crux of the study that we did on these mutations. So then, after creating various mutants of the structure, we performed MD simulation to see what has been the effect of these mutations on the overall stability of the protein. We performed various analysis like RMSD analysis. We analyzed various RG parameters, surface area analysis, RMSF, hydrogen bonding patterns, and PCA analysis and FEL analysis, how stable was the protein during the MD simulation. Again, this is for HR1 mutations. Same sets of analysis was done for the HR1 mutants also. And then we studied same parameters, FEL, IGEN, or PC analysis, RMSF, and so on. So what we found is that, in general, all these mutations were providing destabilization of the protein. And we have seen that the hydrogen bonding profile was also changed because of the mutations. We also found alteration in conformational dynamics, energy frustration in residues, different kind of motions in the protein as the mutants were studied during the MD simulation. Our MD simulation confirmed the destabilizing effect of the mutations, which change its properties and alter the binding affinity against ACE2 of human, which can potentially affect the efficiency of viral entry to the host. So these are our findings, that upon mutations, the stability of the protein is changed, so that can greatly affect its interaction with the host ACE2 receptor, and that can also lead to different kind of infection dynamics in the emerging strains. So similarly, we also studied various other mutants, and we also found the same thing, that the mutations were largely affecting the conformational stability of the protein, and it was affecting its interaction with various intradomains as well as its interaction with the host receptor. So, and then since malaria group is a very large group at ICGEB, and they are working extensively on various high throughput compound libraries which are found to inhibit malaria parasite. And we also were aware that many drug repurposing efforts were on. So we thought of exploring various known antimalarials as antivirals by performing an in silico experiment where we docked the known malaria inhibitors against the main protease of the virus, which is a very important protein for virus replication, as I told you earlier. So in ICGB, we have a set of compound library called MMV malaria box, which is a set of around 400 compounds, which are known active compounds against malaria, but we wanted to explore whether these can also inhibit the virus by attaching to its main protease. And then we screened known inhibitors also using in silico techniques, using a known inhibitor called N3 inhibitor, which is a peptide. And we also docked boceprevir, which is also a known drug to bind to the viral MPRO. And then we compared all these 400 compounds, which of these compounds is able to bind to the MPRO protein better than these N3 and boceprevir drug molecules, which are already known to bind to the viral main protease. And this is the study workflow, that we compared our docking with reference compounds N3 and boceprevir. We performed virtual screening, docking, and then whatever best compounds were there to look at their stability, we performed MD simulations. And then we also performed ADMET and other calculations to see whether these can be good drug molecules to work as potential antagonists for the MPRO. So this is just to show the 5 best compounds which dock very well with the Mpro protein and their comparison with known inhibitors. One is N3, one is boceprevir, as I told you earlier. What were the binding scores of these compounds with respect to the known inhibitors? So these were 5 compounds were the best as compared to the reference compound. So we decided to work closely on these. And we found that the 5 compounds were mimicking the same binding pattern as the co-crystallized inhibitors of SARS-CoV-2 Mpro or N3 and boceprevir. We also found that there were additional hydrogen bonds with these residues, which strengthened the binding between the drug molecules and Mpro residues. So this is which was making their binding better than these 2 compounds. The docking results suggested that compounds have an edge over Mpro, N3 and boceprevir complexes, which is attributable to stronger binding abilities and may hinder the substrate accessibilities. So then we did other analysis of these compounds like calculation of ADMET properties and whether they follow the Lipinski's rule of 5 parameters or not. And upon doing all the analysis, we found that out of these 5, 4 drugs were more better and they passed all the filters, and these can be used for their stability of the complex by performing MD simulation. So we performed MD simulation of these compounds in complex with Mpro to look at their stability. So all these compounds were quite stable. And we also found that their energy parameters were also reflective of their stability in complex with the Mpro. And however, out of the 4, 3 compounds were more better than one of the compounds. So these 3 compounds were shortlisted to be potential Mpro inhibitors as better than the reference compounds that we studied in this work. So this work, which I have shown so far, is already published. Now I move over to the other part of the talk, which is identification of novel SARS-CoV-2 drug targets by host microRNAs and transcription factor co-regulatory interaction networks. So in this work, we used network biology to study the interactions of various virus and host proteins, how they interact. And then by analyzing these networks, we tried to find out various hubs in these networks, which can be probable drug targets. So basically, we looked at the data set, which was published by various researchers. So we looked at the experimental data set of various SARS-CoV-2 genes which interact with host genes. We constructed various regulatory networks, like we generated a network of microRNAs which target the host proteins upon infection, which are the microRNAs amongst these targets which interact with various transcription factors. And then we also compared the various microRNA targeted proteins, whether they are in a gene regulatory network or not. So all these 3 networks were generated, and we looked at the genes which are most connected ones so that they can be good drug targets. So for example, by looking at the microRNA target network, we found that in the host, 2,197 microRNAs are interacting with 311 host genes upon infection. And amongst these, we tried to look at the literature. So how many of these microRNAs are already known as antiviral microRNAs in various other diseases, other viral diseases? And we found that Indeed, 38 microRNAs were already reported to be antiviral microRNAs, and their targets were also known. So they are targeting around 143 host genes. And then we looked at the expression level of these targets in various respiratory infections, and we found that 6 antiviral microRNAs were differentially regulated in various respiratory infections. So hence we label them as potential microRNAs, which can be explored as drug targets, because these are dysregulated in various respiratory infections. So we did a gene set enrichment of various microRNAs which are known to target various host proteins in viral infection, SARS-CoV-2 infection, and we found that many of them are strongly correlated to what could be expected in an infection. So most of the targets are related to apoptosis, cell death, cell cycle, cell proliferation, etc. Similarly, these microRNAs were shortlisted based on the number of targets they have in the viral infection. So these snapshots just to show you that various networks were generated to analyze. For example, this A is a microRNA target interactome, and B is a subnetwork of reported antiviral microRNAs. That is, we found strong evidence of around 38 microRNAs targeting various host genes. So that we plotted. And C is a microRNAs targeting various transcription factors. And D is the gene regulatory network of transcription factors targeting various host genes. So we analyze all these networks, look for the hub genes, and we label them as probable drug targets. Their targets as probable novel drug targets for SARS-CoV-2. So this is a summary of the findings, and I will not read out these details, but we found a couple of microRNAs which were already reported to be antiviral microRNAs. And we also found that one of the microRNA which we found in our study was recently reported to target the 3-UTR of angiotensin converting enzyme 2. And another microRNA was reported to have a regulatory role in ACE2 signaling way back in 2015. The scientists have shown that this microRNA has a role in regulating ACE2 signaling. So there is a strong correlation between microRNA HSA-MADE27B3P and ACE2 is established, but this can be further validated by various experiments, and their targets can be novel drug targets for SARS-CoV-2. So this work was also published. And then I move over to the last part of my talk, where we have developed a machine learning based prediction method for labeling a query compound as anti-SARS-CoV-2 compound. So in this server, we have trained known compounds which have anti-SARS-CoV-2 activity. We studied their molecular parameters, for example, their molecular descriptors, 2-dimensional descriptors, 3-dimensional descriptors. So all these properties of the compounds which are already known antivirus were calculated. And then we also had information about their activity. For example, at what concentration they are producing how much inhibition in viral growth. All this information was already there. So we use all this information to train various machine learning algorithms, and we optimize the models to efficiently predict an input molecule as anti-SARS-CoV-2. So for example, in this server, people can make their own structure as input, or they can post structure in various acceptable formats like mol, SDF, and mol2. And after submission of your input molecule, various descriptors are calculated and various other unwanted things chemical entities are removed, and then it is fed into our prediction method, and then it labels the input molecules as having various antiviral activities, like whether it has potential to be anti spike ACE2 PPI inhibition, whether it shows activity for spike ACE2 PPI inhibition by different another assay system. So since we use various assays for training the algorithm, so the algorithm also predicts the potential of a molecule to be an inhibitor based on that particular assay. So another assay system was based on TMP-RSS enzymatic activity. So whether the input molecule can be a potential inhibitor of this activity or not is predicted by the server. So in red means it is not, and green means it will be showing a good activity against that activity assay. Similarly, we also trained our machine learning model on HEC293 cell line toxicity assay. So the method also predicts whether an input model can be having a cell line toxicity or not. So for example, this one is green, so it means that it is not predicted to have any toxicity against HEC293 cell line. So this is just to show the development of the model. We collected our data from NCAT's assay website. We collected various assays for which complete information of various compounds and their chemical structures along with concentration and activity was enumerated. We collected the data set. We discarded incomplete data. We calculated various parameters of the molecules, including 1D, 2D descriptors. So these are all chemical descriptors which describe a given input molecule, which is calculated using a free software called Paddle. And then we removed all the duplicate entries, because duplicate entries in machine learning can overfit a model. So each entry for machine learning should be unique, and also it may be unique, but it may not be consequential for making a good model. So in machine learning, what we do is we select the features which are very important for classification. For example, a particular property, a particular hydrogen bond, a particular chemical group can be very important for any antiviral activity. So what are those features? So these are done by using various algorithms called feature selection, and they are available free in a machine learning package called Beka, which is also free. And after removing all the duplicate entries, removing all the features which are not so important, then we trained the machine learning algorithms using 80% compounds as training data set. This is done to make or optimize a given model. So only 80% compounds are used. So when you feed 80% compound for the training itself divides it into subsets and then tries to optimize the best model. And after best model is made, from the rest of the 20% compound, what we perform is known as external validation. So external validation is part of the training data set, which is not used for developing a machine learning model. So after looking at the various performance parameters of the external validation data set, the best model is selected, and then we put it up as the prediction model on the web server or standalone version of your machine learning algorithm. So this is how Scope Thread was developed, and it is available as a web server as well as standalone, because many scientists don't wish to share their compound library on web. So we realized this requirement of the scientists, and we developed a standalone version also, which is also very easy to use. So this is how the server works. You input a molecule, the Scope Thread server will accept the molecule, calculate its property, and then it will run the query descriptors and input file using the best ML models that we have selected in this step. And then it is giving the output of the prediction for these assays for which we have developed the models. So this model is helping us predicting the compounds which have potentially anti-SARS-CoV-2 and which also don't have any toxicity against human cells. So this is also one of the uniqueness of this server, that not only we are predicting the antiviral compounds, we are also predicting whether they can be toxic to human cells or not. So this web server is already online and it is currently under review for publication. So these are some of the publications from my group since the onset of the pandemic. So many of my students were sick with COVID-19, but even then they were working, and a few of them also lost their relatives, but that did not stop them from working in the area. And they were able to put up some research work, and which we were able to predict, and hopefully they will be useful to the research community. So this is my last slide, where I wish to acknowledge the contributions of my students: Shazev Ahmed, Hema, Shweta Birla, Neeraj, Arun, Rahila, and Deepshika. And I also acknowledge the funding from Department of Biotechnology, Department of Science and Technology, CSIR, ICMR, and few of the illustrations were created with BioRender. So thank you very much for your attention. Stay safe. Thank you. I can't hear you.
Hello, sir.
Yeah, hello. Now I can hear you.
Okay, so Dr. Ramneet, you may proceed.
Dr. Avneet, you are not audible. Please check your connection.
Okay, Dr. Ramneet is asking that can we take few questions from participant, because they put on some discussion questions. So with permission of Dr. Dinesh Gupta, we can take it.
Yeah, please go ahead.
Yes, sir. Thank you.
So the first question is from Mr. Abhishek Chahar. He's saying that he has done a project in his bachelor's degree where he has done molecular docking of COVID proteins with various phytoactive compounds and has also seen some good results. But he's asking that can he publish his work without any experimental validation? Will it be possible for him?
Yes, yes, why not? I just showed you that we have published 3 publications without any experimental validation. So you may not get a publication in a high impact journal, but there are journals which will be accepting such papers also.
Okay, sir. Thank you. Abhishek, I think your query is, the answer is clear to you. So the second question is from the other participant. She's asking, what will be the basic requirement to understand machine learning techniques?
Your voice is feeble. I can't make out.
So the next question is from another participant. She's asking, what will be the basic requirement to understand?
No, your voice is gone. I can't hear you.
Dinesh sir,
Dr.
please
Ravi,
allow
can you
me.
please
Yeah,
continue?
I'll continue, Ravneet ma'am. So the next question is by Manjusha Sudhakar, and she is asking, what will be the basic requirement to understand machine learning techniques, whether we need to complete the course in AI? I think she means artificial intelligence.
Okay, okay. For machine learning, there is no need to have very expertise. You can access the machine learning tools like Beka, which are free to download for any operating system. And there are very good tutorials on the website, which you can follow and learn. And then you can run your own machine learning experiments. So it's, if you are you have a basic understanding in how to run different software, so it should be fairly very easy for you to pick up.
Okay, thank you, sir. Thank you for answering the query. Manjusha, I suppose your query is resolved. So we have another question from Miss Aditi Nag, and she is asking, so how can I compare the spike structure that I have modeled with the structured reported on GISAID?
So GISAID has many structures which you want to compare it with the wild type, then you have to download the wild type structure and then superimpose your model to it, and then you can compare. You can use software like PyMol or Chimera to compare the structures.
Okay, so I guess Aditi, your query is also resolved. Yes, she has written okay, so thank you. So the next question is from Mr. Mohammed Jamaluddin. He is asking, so what is the main object on the MProServe attractive target in viral replication?
So MPRO protein is actually required in processing of ORFs of the virus into smaller proteins and peptides, which are important for viral growth. So that is why NPRO is one of the attractive targets for inhibiting the viral replication. So that's why people are researching on how to inhibit the MPRO activity.
Okay. So participants, if you have any more questions, you are requested to put up the same in the chat box. Ravneet ma'am, over to you.
Okay, ma'am. Thank you so much. So I hope no questions are there from the participants now. Just last question has just come. So this question is, does the study on miRNA is the final solution to find out the effective drug against virus?
No. miRNA studies are just to find novel drug targets. So the targets could be either in the virus or in the host protein, because host proteins are also giving so much effect on the host, that much of the activity that you see on patients is due to the host interaction with the viral proteins. For example, the immune system is completely overwhelmed in cases of virus infection. So whether host proteins can be used as drug target or not, that we can know from our studies on microRNA host protein interactions. So microRNA may not be the effective tool, but it is a tool to find out various drug targets, either on the host or on the virus.
Thank you so much, sir. And the participants are very happy with your explanations. They're also typing in the chat box for such a wonderful session. So I would thank you so much, sir. I would now request our Honorable Director and Dean Research, Professor Vinay Sharma, sir, to kindly grace the session with his few kind words.
Nice to see you, Professor Sharma, after so many years.
Yes, Gupta. It's a pleasure, a great pleasure for me to see you after a long time. I think
Same here, sir. Pleasure is all mine.
because we had, DBT, that coordinators activity has closed now, Center for Bioinformatics. I think that was a very good opportunity for every one of us to interact. So I think it's indeed, I would once again would like to extend my wholehearted welcome to Dr. Dinesh Gupta, who is a pioneering bioinformatician of India. And, I joined the session late, but, he has contributed a lot. Though I was towards the end of the session, I could make out he has contributed a lot to the contemporary problem of COVID-19, And these were difficult times, and difficult times are still continuing. And despite all those bottlenecks, despite all those difficulties, he has been able to carry out some landmark work in the area of COVID-19. And as he told that many of his PhD scholars, they also got infected, and laboratory also everywhere situation has been same. The laboratory workers also, technicians and so on, they got infected. But I think his just for zeal for work and making novel contributions and making something new to overcome this crisis to our nation, to country as a whole in the time of COVID-19 is really, really highly praiseworthy. Dr. Dinesh Gupta is famous, as I've already told you, and he sits in many, many task forces also in the DBT Bioinformatics Committee as well. He sits there. And it's indeed, once again, I would say it's a matter of great pleasure for all of us that he could find time out for us for this 2-day workshop. And I'm very sure that all the participants must have been very, very immensely benefited by his contributions. So thanks a lot once again, Dr. Dinesh Gupta. Thank you.
Thank you, Professor Sharma. Thanks for your kind words.
Thank you, sir. That was a very befitting closure by Professor Vinay Sharma. And I would also like to thank Dr. Dinesh Gupta for a lucid explanation for studying the SARS-CoV-2 virus. I'm sure the participants will be actively looking at all the databases and the sites you have mentioned in your lecture. And we might be getting a lot of queries post the session, and we hope to continue questioning you for clearing our doubts. Thank you, sir. Thank you so much.
Welcome. Thank you.
I would urge the participants to keep in touch with him. They are going to learn a lot from him, in bioinformatics.
Sure, always welcome. Thank you.
I may please request the participants to stay back. The participants, there are a few instructions for you. First of all, you might have received a mail late at night yesterday for installation of an program, Autodoc. I hope for the next session, you guys have installed the program in your laptop, because it will be a hands-on demonstration for the same. And the session will start at 2 o'clock sharp post lunch. So I hope there will be a timely attendance, because if you miss something, you will be at a loss. So hope to see you soon. Have a great lunchtime.
Good afternoon. Sir, actually,
Yes,
our participants are going to join in a while.
okay. That's
So
fine.
we'll just wait for next 5 minutes and then we'll start the session.
Sure. No, thank you.
Ravneet, ma'am.
Yes, ma'am.
Ma'am, you will have to, once the participants have joined in, you have to request them to please turn on their cameras at the end of today's session so that a group photo can be taken of everyone. The camera rights will be given to them at that particular time.
Okay,
Hello,
ma'am.
Dr. Puneet.
Definitely.
Yeah.
Okay, so this is a practice session. So you may share your PPT and just check whether it is working or not.
Okay,
So
sure.
just
Very good.
I think you are already aware about Zoom, but still, I mean, you can check it.
Okay, okay.
Once again, I request all the participants to kindly inform your friends to join the session so that we can start without wasting the time.
So I'm sharing my PPT.
Yeah, right.
Okay. Can you see this?
Yes, yes, yes.
Yeah.
Sir, you can
Sorry.
unshare.
Ah.
It's very clear.
Okay.
So Dr. Manishita, 47 around participants are there. So can we start the session now?
Ma'am, just give 5 more minutes. We can wait till 2:05, then we can start.
Okay, fine.
Right. Hello, Dr. Puneet. Can you hear us?
Yes, I can hear you.
Okay, Dr. Ramneet, you may proceed.
Okay, sir. So Dr. Manishita, can we start now?
Yes, ma'am, you can.
Okay, ma'am. So good afternoon, one and all present here. Once again, I, Dr. Ravneet, on behalf of Organizing Committee GBE, welcome you all in the second technical session of this 2 days online workshop on bioinformatics and drug engineering, organized by Amity Institute of Biotechnology, Amity University, Rajasthan, in association with IEEE Rajasthan subsection. This technical session will be the hands-on workshop for which we have very esteemed scientists amongst us. So now I take the pleasure of introducing Dr. Puneet Singh. Dr. Puneet K. Singh received his MSc degree in bioinformatics in 2011 from BIT Mesra, Ranchi, and his PhD is in microbiology from Maharshi Dayanand University, Rohtak. His current research interests are system biology, protein engineering, and peptide production. His research publications have endured him with 654 citations in a very short span of time, with a very higher H index of 17 and I-10 index of 20. He has been awarded the Young Scientist Award by the Association of Microbiologists of India in 2016 for the study of inulina's application in food industries and health applications. He has worked as a visiting scientist at Stage Key Laboratory of Pulp and Paper Engineering, South China University of Technology, Guangzhou in China from 2017 to 2019. He was later appointed postdoctoral scientist at Vaccine Private Limited Company, running in the Department of Endocrinology at Flinders Medical Center. Vaccine is well known for its advex range of vaccine adjuvants implemented with influenza, therapeutic hepatitis B, and therapeutic allergy vaccines. Vaccine is also the first company in Australia to enter human trials for its vaccine against COVID-19. He has also worked on a project funded by the National Institute of Health of United States government. His work at the vaccine consists of molecular modeling, molecular dynamic simulation, and also the virtual screening. He did a project related to nucleotide binding oligomerization domain containing protein 2, that is not 2. He is currently working as a postdoc at Onam National University, Gwangju, South Korea. So now with these few words, I would now hand over the session to Dr. Puneet Singh.
Thank you, Dr. Ravneet, for this introduction. And let's start the workshop so that the participants should get an hands-on on the molecular docking. So just I am sharing a PPT. It's just a short PPT describing what is molecular docking, and there are some key terms, and so that you know they get to know actually like what docking is, and like before performing, you know, they have an idea. So yes. So what actually docking is docking is a method which predicts the preferred orientation of one molecule to a second when they are bound to each other and they form a stable complex. So like we have a protein and we have a ligand, and we want to see it like how they are binding and whether the binding is very strong or not. So and where the interaction is taking place. So there are two approaches that are very popular with the docking community. Is like one approach, which is like just we are matching the surface of the ligand and the protein. And the second approach is like it actually simulates the docking process, like what are the energies and what are the interactions between the protein and ligand, and what are the bonds forming, and you know the breakage of the bonds, and finally what is the energy, so that we can compare between like if there are many ligands and just one protein, so we can see that which ligand is effectively binding to the protein. So to perform docking, we need to have like PDB structures, that is X-ray crystallography or NMR structure that is present in the PDB website. And so in that, you know, if the more clear the structure is, like there is a resolution in Armstrongs, so that is better. And we can download the ligands from like different database like PubChem, drug bank, etc. And also you can draw, if your ligand is small, you can draw in CamSketch and download it in a PDB format and then proceed with the docking. So there are different terms. I think if you can see, so it will be much easier if we'll talk later upon. So there's receptor. Receptor is just the protein. Mainly the protein is the receptor. And the next one is the ligand. Ligand is any chemical structure that inhibits a protein or propagates its activity or anything that binds to protein. And docking is the computation simulation of ligand and receptor. Binding mode is how the ligand is binding to the protein, the orientation of that. And the pose is also like when we study the the complex, we say that how the ligand has binded to the protein and what is its pose. So it's just a simple term. And scoring is like we do a scoring in docking actually. So there is a value after docking. That is the main criteria while comparing with different ligands. So scoring is basically calculated on the formula, depends upon the like how many hydrogen bonds, how many hydrophobic interactions, and there are a lot of parameters that are taken into account. And the next ranking is the process of classifying which ligands it's most likely to interact favorably in this with the receptors, and then we calculate the free energies. Like these are the just few terms, and there are a lot of tools already available in the web, and like you can see, there are different website. So in this session, we'll be working on the AutoDoc. So I think Dr. Ravi has shared you a few links, and please let me know if you have downloaded that. So if every participants have downloaded those softwares and installed it, and if they have, okay.
Sorry to interrupt, Dr. Puneet.
Yeah.
Although we already mailed them instruction for downloading AutoDoc and file that we will use in AutoDoc, right?
Mm-hmm.
But I recommend to give them 5 to 10 minutes just for installation and getting used to environment of AutoDoc, right?
Okay.
So that they can arrange that PDB and ligand file, receptor and ligand file. And if anybody not downloaded it, so they can download in next 5 minutes or maybe 5 to 10 minutes.
Okay,
right?
okay. So let me tell you, like, there are two files. One is MGL tools for the Windows, and the other one is AutoDoc. So two installation you have to make. The AutoDoc, is very small. It's like 567 KB something, and the MGL tool is like in 70 MB something. So there are two files you need to install. Okay, then we can proceed with the other things.
Yes,
So
and first you have to install MGL tool, because AutoDoc only will run after MGL tool, right? If I'm not wrong.
Yeah, right. MGL is the main one, and then okay. You can you can take 5 minutes maybe. That will be great. If there is any problem, you could just come to me.
Right. In between, someone from participant asked about link. So just I'm copying here in chat box. Yeah.
Okay, you have sent the link.
Already I mailed, but I will copy here the link in the chat box, right?
That's okay.
Ravi sir, all the participants would have got the email second time also. I had sent at 1:30 also. The participants are requested to please check their emails.
Okay.
I think this someone is asking for the Mac. Actually,
Mac,
there are
Mac
yeah.
OS file. All right.
Yeah, Mac OS
Yeah,
file,
they,
someone is asking.
yeah, they can find it over there. There's Linux file, Windows file, as well as Mac.
Yes.
I,
At that
I,
particular.
let me see if Autodoc is in Mac. Just 1 second. I think it's there for Mac. For Mac, Windows, and Linux, everything is there. You can download it.
Yes, MGL tool is available for Mac also, and Autodoc also available. I think just follow the link. The second number
Ravi
from
Ranjan
that.
sir, can you please share the screen so that the participants get to know from where they have to take what?
Yeah,
Okay,
that's it.
You can just share the screen and show them, sir.
I'm sharing that screen.
Okay,
Okay.
sir.
So this is the screen, MGL tools, and below this there are installers. So as you can see, MGL tools, and here is a sign for Windows, for Mac, and for Linux. They can choose. I think it's okay. So are they getting any problem with Mac, like regarding installation? Because they have to untar this one. I think everybody has seen this. It's fine.
Yes.
Yes,
Yeah,
Puneet sir, you can unshare the screen. I guess
Okay.
now it's fine.
And I request you, Dr. Puneet, to now begins with demonstration in between participant, because participants are already informed about the installation. So I
Okay,
don't think they need much time for that, because further we need time for analysis and hands-on.
So just 1 second. I'm sharing one more screen, like the second link, what they need to do, and then after that,
Puneet
I'll
sir, just sorry to disturb you.
Yeah.
Tonya Kriba, sir has just shown us how to get it for the Mac OS. So if you are not attentive in the session, then we cannot help you with the same. So please be attentive. It's a request to all the participants. Please be attentive and stop bothering us again and again with the same things. We have already shown it to you how the things work for the Mac OS. Okay. Thank you, Puneet sir. You can continue.
Okay. So I'm sharing you the other screen. Is this one? I hope you have sent. This is the Autodoc one. The previous one was the MGL tool. So here you can download the Autodoc and install it. Okay. So
just one thing for participant, Dr. Puneet. Listen, all participants. It is most obvious when we are working on some software that maybe depend on system to system, maybe not work, or it take longer time. So don't worry about that. Just focus on the demonstration. And if any one of you, I'm not saying that all of you, but maybe one or two participants face some problem of running the things. So don't worry about that. Just watch the demonstration, and we are available for you even after that workshop. If suppose 1% or 2% participant facing some problem, because it is most obvious. I think Puneet, I'm right that
yeah. Because also
it's happened many times with software. that depends on system. Maybe it work and maybe it take longer time, right? So that is considerable, right?
Yeah,
Okay,
and
Puneet.
also,
Okay.
if you have something like any problem, you can, ask afterwards as well. So I'll be available, no problem. So because it's very difficult to explain everything in like 2 hours, but it is just a demonstration so that you can have an hands-on and you can run and you can get the confidence, like if somewhere you can say that you have done docking and you will be fine after this one. But let's hope if something if there is any problem, you can just come to us, no problem. So after installation, just 1 second. Mhm. Just give me 2 minutes, I'm just finding a file. Okay, so when you have installed both the Autodock, MGL tools and Autodock, so here's the location, as you can see in the screen, like this PC, operating system, program files, the Scripps Research Institute, Autodock 4.2.6. So can you go to this location on your computer? Just take 2 to 5 minutes, no problem. You need to find these two files. I tried to share it, but it is difficult to attach extension files in email. So
Yeah, right. Extension file cannot be sent through email, and
wait,
maybe it's a security
so it's
reason.
it's better, even if you can't find, you can run the Autodock, but it's better if you get these files because then we have a lot of control in our hands.
Okay, Dr. Puneet, we received many of the messages that they find it in their PC.
Okay, fine. Just copy these two files. If you find, whoever have find, just copy these files. And as I've made just make a folder and paste in this. Like I have a lot of files in this one. Just keep one protein, which Dr. Ravi has sent you, and one ligand, and these two files. So here you can see is like these two files are here. I hope you can see the screen, like whatever I'm doing. Dr. Ravi, that's okay. You're mute, I think.
So it's clearly visible.
Okay, that's good.
Yes,
So just-
Dr. Puneet, your screen is visible.
Yeah, okay. So these two Autodoc 4, Autogrid 4, these two files, and one ligand file and one protein file. This Dr. Ravi and from the Amity group you must have received. So just paste in this folder, one folder, just make up one folder and paste all the four things. Like these two files and one ligand file in the PDB format and one like protein format. Just the other files you will get after docking. So no problem.
So to the participant, actually, we are providing a similar path for the receptor file and ligand file along with the installer, right? So we are making a file by the name of workshop or maybe any project of you and make it on desktop. And so that we have the similar path of ligand receptor file as well as installer, like there's two file, Autodoc and Autogrid. Right, Dr. Puneet?
Yeah.
Yeah.
So like so like I can make yes.
Ravi sir, sir, 2-3 of the participants are calling me and asking that the file is not opening. So what?
I have to open it here.
Ravi sir, Dharmsheel is asking a question how to get other two files. And Krunal is saying I am unable to locate these two Autodoc in Ubuntu Linux.
Okay.
So for Ubuntu,
Ubuntu, I think
for Ubuntu, if they are not locating Dr. Puneet, then it's fine. Their file will be generated there by using their path.
I think so. Just try once because if that is installed in that, so maybe it will take automatically. If it doesn't take,
And
then let me see what is the problem here. So let's start once and see if Ubuntu sometimes take automatically. It's not a problem.
yes, Abhishek Chahar, most probably you will get it automatically because you are asking about Ubuntu. And if you not find, then you may check the path of that particular folder where Autodoc is installed, right? So we are doing all this for path only, right? There is nothing special. So if you're working on Ubuntu, so I think already you understand the path things of path of any file or folder, right?
Ligand files, I think someone is asking.
Yeah, thank you.
Ligand files, I think you have sent
Yes,
by
yes,
mail.
I send it. Okay, Puneet,
Sandra,
so,
the ligand files are in your email. Check your email, which you must have received from my end.
So there are 4 files. Now it's much clearer. Just 4 files you need to have. Okay, now I think should open. So just type in the search bar Autodoc tools, whoever is using with this Windows 1.
okay, Dr. Puneet.
Yeah.
So I think you should go with demonstration because we
I'm
have
just
a
starting.
time-bound session also.
Yeah.
So, and if anybody is having problem in installation or something else, so they maybe interact after the demonstration, right?
Okay.
So I request all of the participants to observe demonstration and then try it along with. If you are failing to do so, then maybe you try next time. And
someone is asking downloaded file is not opening. Which file is not opening because there are 2? So just mention which file is not, the MGL one is not opening or the other one. Just let us know, and also your operating system. So this is the Autodoc. I'm just opening just the Windows people. You can search it on the bar.
Yes.
And so there is a trick in like Linux one. I think if whoever is working on the Linux one, they need to give a path, and you have to type like ADT tools in the command line and then enter. So if the path is correct, you will get like this screen, and your Autodoc will open automatically. So here in Windows, you just type in the search bar, and after that, remind me later. There's like they want you to register for something. So if you have time, but now avoid it. So here's the Autodoc tool. So I'm just starting it. Okay. So first of all, you need to open the protein files, and this is read molecule. Okay, so molecule is in like desktop, and I've created a new workshop folders, and 6M71 is the protein. Okay, so I'm opening it. Okay, this one. So let me tell you one thing, how you can find the protein. So it's the so here you can use the RCSB PDB. It's the PDB homepage. So for example, I'm just 6M71 is the protein, or you can just write the name or anything, or you'll get a lot of options, and you'll get to know what you need to download. So after clicking this is our protein. Okay, and here what you do is just go to these download files, and this is the PDB format. And once you click, you'll that will be downloaded here.
Dr. Puneet, probably your screen is freezed. So can you check it?
So which screen you are able to see is the PDB one?
We are seeing that 4 file, that is PDB, Autodoc, AutoGrid, and ligand file is in a single folder.
Okay.
Yeah.
Okay, can you see now?
Yes, now we can see.
Okay. So you have to go to RCSB PDB homepage and click on the homepage, and in the search bar you can click 6M71. You can also write the protein name or any other information you have, and you want to download some protein, and just search it. And then there is this one, and then go to download files, PDB format, and here as you can see at the down left hand side, your protein is downloaded. So this is how I have downloaded this protein. And also you need to trim some of the protein. You have to go some of the visualizer tool to open this protein and see if there are other things, because these are X-ray crystallography or NMR structures, so they are not very pure. So you have to take out all the impurities, like sometimes there are metals, there are water molecules and other things, so you can remove, and which I've already done, and I've sent you the protein structure. So so here is the Autodoc, and what I have done is file, and then read molecules. And I have chosen the protein which already was in the folder, and I have opened this protein. I think you missed because my screen was frozen. So once you have opened the protein, just 1 second. Okay, someone is asking how to. So there is a tool called Chimera. So that you need to, that is a visualizer tool. And if you want, I can show you. So this is the tool, and like I'm showing you how I did. Like you can also download fetch PDB from here 6M71, and like you can just wait for a while till it downloads. So here is your protein in Chimera. So someone was asking how to take out the impurities. So the first thing is you should know everything about your protein from the literatures. Like you should know what protein you need to have and what chain you want to have, what is your active sites. These all information you should get from the literature. So it's not that it is just docking. So you can take any protein, any ligand, and just go on blind docking. So you need to have a specific, the site where you can bind your ligand. So here I can show you. So here are like there are 4 chains, as you can see on the left side: A, B, C, and D. So you should know like which chain you need. So like if I need uh, A chain, like this is the A chain, okay, and the rest I don't need. So what I do, I just go and I'll do invert. So except for the A model, I'll remove all the chains. So I'll go to atom bonds and delete. So it's deleted. So likewise, if there are any impurities, you just go to select, and there are residues, whichever residues are there, it will be shown over here, and you can just select and delete it. That's all. And you can save this PDB and proceed with the docking. Okay. So I'm just closing this one. Okay, sorry. So again, coming back to this autodock. So once you have the protein, so you need to go to the edit section and delete water. If else water is there, you can also prepare your protein over here. So delete water. Don't worry if you miss any step. We'll be providing you with a all written step-by-step things. So don't worry about that. Okay.
And even recorded session of this technical session on Autodoc is available on YouTube. So don't worry about the things that you missed maybe.
Okay, so what we'll do is, okay, so we have already done the delete water, and hydrogens is add. Just do it the default one, no problem. Everything will be fine. So, okay. So you have deleted the water, you have added the hydrogen, this one, and then go to file and save this PDB. So while saving the PDB, just be careful like the path of the file name, because in my system it takes automatically something else. So I'll go to desktop, and then here is workshop new, and my file name is 6M71. Just replace it with the new file, no problem. So I'm replacing it, and just take this all the 3 checkbox, sort nodes, save transform coordinates, write all bonds as connected records. Okay, and just say okay. So there is already a file. So yes, we want to overwrite that. Okay. So I'm just waiting for 10 seconds that you guys are doing. Okay, so after this one, so let's come to the ligand. This is your protein. You have saved the PDB coordinates. Okay, just come to ligand part. So ligand, then input, and then open file. Okay. So my file is desktop. Workshop new. So you can't see any file because it's showing the PDB Qt format. Just changes to PDB files. Okay, just if you don't find any file, just look at the in the site drop down, like which file it's searching for, and here we have the ligand, and then we have opened the ligand. So here is our ligand. Okay, so ligand is done, and then we have introduced the ligand and just come to our output and save the ligand as PDB. So PDB Qt format is very necessary for Autodoc because Autodocs only takes PDB Qt formats for the docking. So now what we have is a PDB format. So what we are doing now is converting the protein and the ligand into PDB format. So ligand workshop new, and then save. Okay, so you have saved the ligand. Now what you have to do is, so you have protein, you have ligand. Now just come to grid. So you have to make a grid, like what are the areas where you want to your ligand to bind the protein. So it should be very clear. So here in the left, because this protein is not very clear, if this is your protein, it is difficult to find out like which is your binding site and how can you make the grid. So I'll show you once after this grid one. So come to grid one, okay, and then choose the macromolecule. Okay, so macromolecule is your protein. Open, like you can choose because already it is there. So 6 and 7 1 is your macromolecule. Select molecules. Okay. So once you choose the macromolecules, it will automatically give you a window in which you can save this. As you can see, save the protein in the PDB file format. So this is a little bit critical step. So rather than opening the macromolecules, you have to choose the macromolecules. So go into this one, and now you have already you have a ligand in the PDB Qt format, as here you can see, and now you have the protein in your PDB Qt format. So it's done. Okay, so both the things are saved in PDB Qt format. Okay. Now what we have to do is, okay, so come to grid again and set my map type. So in set map type, there are a lot of options. So in this also you have to choose ligand, like you did on the previous one. So here is the ligand, LID remdesivir, and select ligand. So you have selected ligand. Next, come to grid. So when you come to like grid docking and all that, you have to systematically go one option by one. Okay, so we have done macromolecules, set map type, and then come to grid box. So I think you can see the grid box over here. So because it is very difficult in this one to find out like what are your sites, and which you have to choose, because you just have to see into the protein and like make an assumption that this should be the site. For this one, you can here in the left hand side, as you can see, there are like 6 and 7 one. I think if you are able to see the cursor, so you just click on the ribbon model. This is R. Just go to this one, click here. And just unclick this one. So this is much clear, and you can choose what are your active sites. You can see like where are the ribbons, where are the sheets. You can go and make an assumption. So and one more thing, you need to have the exact positions of the ligands, the amino acids where it is binding. So you have to thoroughly study the structures to find out where this box should be there. So this is a by default size of the box, but you can increase it like from here. You can just do it like this, increase here. Okay, so like this one. Okay. And also if you want to move upwards, downwards, anywhere you can move this one. If you want to move like this one, the whole protein will be covered. But I suggest you to be specific as much as possible, because the more you will increase the box size, the more time it will take for the docking, because the more computational energy it will take. So it's better to be more specific, and you'll also get the more specific results. Like I'm keeping it a little bit small, okay, for now. And I'm not changing, I'm just keeping it as default as it is. After this one, go to files and do close saving current. Okay, then again go to grid and just go to output and save the GPF. GPF is the grid parameter file. So you have made a grid, and all the information which you have, like you have increased the box size and everything, what is a protein, ligand, everything will be saved from this one. I'll show you. So it will be saved. So this is a very crucial step as well. So just go to desktop and workshop new, and just I'm giving rem dot gpf. Don't forget to write the extension. So just rem because the ligand name is remdesivir, so I'm keeping it as rem. The shorter the better it is. Okay, and it is saving in .gpf file, but you should write .gpf and just save it. Okay, so let me show you what was there in the GPF file. So you can see in the GPF file, and these are the points, the coordinates, and what are the spacing, the receptor information, what files will be generated, the grid center, as you can see, the receptor, and all information is there in this one. Okay, so that is saved. Now come to docking. So grid, we have made a grid, And now we are coming to the docking one. Here, we come to macromolecules and set rigid file name. Rigid file name because we are keeping the protein as rigid one. We are not allowing it protein to move, just we are allowing the ligand to move. So in this one, we are opening. And so macromolecules is protein. So just we are opening this. Okay, selected docking. After molecules, ligand. Ligand you can choose because it's already there. So ligand remdesivir is there. Okay, they will ask you some parameters, and just you can choose random. Sorry, it's like by default, no problem. And then again go to docking, search parameters. We are using their genetic algorithm, and here are the number of, like you can change the number of genetic algorithms run, what should be the population size, and how much evaluations do you need. So there are a lot of, just keep it 10 GA runs, and that will be okay, no problem. And then after that, docking parameters, accept, and other options. You don't need to have other options. And then output should be in the genetic algorithm because you have chosen docking parameters for this one. So just go genetic algorithm. Okay. So here is the previous one was the GPA file. It was the grid parameters. Now this is the DPF. It's the docking parameter file. Go to desktop, workshop, and again I'm naming it as REM dot DPF, docking parameters file. So whatever you have chosen the parameters, like the default values or whatever values, all will be saved in this one. So this one is done. So just take 10-20 seconds and then I'll go to the next step. Okay, so now there are two methods to run the docking in this one. Just run AutoGrid, and you can choose the program file name. Like I've said, if you don't find the path name, you can go over there, like this one, and program path name is already, AutoGrid is defined here, and parameters file you can choose from here, and log file name means you have to give a path where your files will be saved. But I'm not doing this way because it's sometimes it works and sometimes it doesn't work. So what I'm doing is going into the command line is cmd. So I can go into the command line prompt, the Windows persons, and as well as the Linux one if they are able to find it. So here is the command line. I'm just going to my the folder, like cd.
Dr. Puneet, your command prompt is not saying, I mean, terminal is not saying.
Okay.
I think you have to minimize Autodoc, then it will be.
I'm sharing just the command, this one.
Yeah, right.
Okay. The command prompt. Now it's fine.
Yeah, I can see.
Okay. Yeah. So here, I'm just going to my folder. So CD desktop and CD, what was the name of that folder? Just one second.
Workshop new.
Workshop new.
Right.
Okay. So we are in the folder. Okay. So first we will run the auto grid and then we will run the Autodoc. So for auto grid, like the two files which I've said you to copy from, the folder to your folder was auto grid 4, right? So auto grid 4. This was the, the tool. So you have to write in this one because already that we are assuming that AutoGrid 4 is there in this folder. So AutoGrid 4 and then minus P. And so you are giving, because this is AutoGrid, so you're giving rem dot grid parameter file GPF, which I have made. Okay. And then minus L rem. So I'm minus L is the output file after the AutoGrid file. What will be the output file? So what I'm doing is rem. I'm giving this name to rem.glg. So glg is grid log file. So there will be, this is a log file containing all the information which will be generated after the AutoGrid is run. Okay, so I'm entering. So it's running. Okay. So let's wait till it runs. It won't take much time. So I think mine is completed, this AutoGrid. And after that, so just go to the folder and so go to the folder. As you can see, I'm sharing this one. So now you have these much of files, okay, all the map files, grid parameters file, grid log file. So this is the rem glg file, which is now, so you can see it's almost 190. And also here in the command line, you can see like it's completed. So, is it fine? So just note down these commands. It's very simple. And so now AutoGrid is completed. Now we'll run Autodoc. Autodoc. Initially it was AutoGrid. So to run these files, it should be there in your folder. That is why I was saying to copy these two files from that. And then minus P. So GPF is done, grid parameters file. And now we'll do rem.dpf. It's a docking parameters file. and minus L, and then rem dot DLG. It's the docking log file. So in before this, in the autogrid we received GLG grid log file. Now we'll receive the DLG. DLG is very important file. It is the final file from where you can do your calculations and all that. So I'm not clicking this one because it will take a lot of time, and I have already done this one, but you can run in your computers. So just click this one, and that will start.
Okay, Dr. Puneet, I request you to demonstrate with that GUI interface also about AutoGrid and
Okay.
AutoDoc run.
Okay, I'll do that, but yeah, but I'm not sure. I think it's
That's
not working in my system. Yeah, but
that's
yeah,
fine.
I'll do that. Yeah,
just that's fine.
just I'm doing here.
So, participant, please note down these commands for AutoGrid and AutoDoc. Maybe Dr. Puneet will copy in the chat box.
Yeah, there is a file I'll send you maybe.
Yeah. Okay.
Yeah, then you can share it.
Okay.
So this is the file, I think. Okay. So you can go to run AutoGrid, and you can see this little window over there. Dr. Ravi, is it visible? This smaller window.
Yes.
Okay, the smaller one is visible. Okay. So you can browse from here, and like I think it's the output file, parameters file. Already the location is given. C uses this desktop workshop. Okay, so it's fine. And the log file name, where you want your log file to be stored. So while browsing log file name, you should write it manually because you want to save that file. So like I'm saying rem1 because already dot glg. So it's the same as the command because you are giving it the GPF file, you're saving glg file. Let's see, I'm not very sure. So you can see like here is a cmd. It's written down here, and is the same command they have written autogrid 4 minus p, the path of the GPF file, and then minus L and path of the this one. Okay, it's the same one. It's a problem. I think there is always problem with finding the path. That is what I'm saying. If someone system is taking the path, that's well and fine. You can try. But what I have shown you is the very straightforward part. It won't go wrong any time. That is what I have shown you. Okay. So I thought you should not confuse with all this. So there is something they didn't find the file. That's the problem. That's okay.
Okay.
Did you get it?
Okay,
Yeah.
Dr. Puneet. So I request you to give 10-minute break to the participant. In this 10 minute, they will try AutoDoc, whatever you discuss in this session, right?
Sure.
And after 10 minutes, you will resume session with analysis part. So is that right? Is that right?
Yeah, that's okay. No problem. Okay.
So and
just we are taking 10 to 15 minutes because depending on the system, someone's will, and maybe they can go back. Yeah.
And if you have steps written over, then you may share in the chat box so that it will be easier for participant.
Okay. Just I just have to find. Meanwhile, I'll find and I'll share. Okay.
Yeah. And to the participant, don't worry if you don't get the result, because we are sharing the steps also, and even with the permission of Dr. Puneet, I assure you that we are available for you in future if you are working on some project or something. So don't worry about it, about that. And it is quite difficult in a single training session, and you get all autodoc steps and all. So maybe take some more practice session. So don't worry about this, but you try whatever we discussed here, and at least you will learn something, right? And then after 10 minutes, Dr. Puneet will resume with analysis part of this autodoc, and also he will take some your query, general discussion kind of thing. So 10 minutes, right?
So yeah. Let's
Yeah.
meet after 10 minutes. I think we should now.
Yes, Dr.
Yeah.
Now, I think we can resume our session.
Yep. So just go to the. Just wait a second. So is anybody able to run the autodoc final? It also depends upon the computational capacity of the system. Sometimes if it's very good, it is very fast. Someone is. Okay, so let's. So I'm sharing my screen. Okay, so are you able to see my screen, this one? And so here are all the files, and here are all the files, after docking. So what you have to do is just open the autodoc again. And so here is the autodoc. So go to analyze, then docking, and then open. So here you can see it is written select docking log file name. So you have to, so the as I've said you earlier, there will be a file generated after this docking, that is .dlg docking log file. So now what we're going to see is like here is this is the previous one I have done previously. This is not for, but this is the same file you will get, just the name is different. So I'll open the .dlg file, and so okay, so there are 50 confirmations. Okay, done. Then analyze. So first we did the docking one, open the DLG file, then go to macromolecules. You can choose it because already it is there, so you can choose. Okay, then go to confirmations and play. You can play it or play rank by energy, so it will be giving you ranking, like which should be at the first position, at the second position. So here you go with this one. Then you can click open the panel. Just click on the and sign here and you'll get this panel. So click on this show info. So you get this information, and so this is the first one. As you can see here is the first confirmation, and because we have done the play by rank of energies. So this is the best pose you'll get, and the binding energy is minus 4.3, and this is in the kilocalories per mole. The unit is. So, and if you click here, there's the second one, is slightly less, so less value, minus 3.78. And as you go ahead, you will get the lower energy because it is in decreasing order. And you can see here the little ligand. It's going to different poses. Okay, so there are 50 conformations. So we don't want 50, but we want the first one because it is having the lowest energy. Or if sometimes what it happens is your ligand might get the best energy, but it is not binding to the site which you want that ligand to bind, because in literature, suppose you have read that this particular site is very important for the binding of this ligand, but you are getting the lowest energy where the binding is not done at that site. So you have to see the FD conformations, and you have to find out like what is the optimal, is it binding to the right site or not. So there are two cases: either you want just the energies and you want to see if it should bind at the with the minimum energy, or it should bind at the right side. So these are two different aspects which you have to take care in this one. So depending upon your requirement, what you have to do, you just choose carefully. So in this, because we are studying, so we'll take the first one, and so you can copy, like there are a lot of informations you might need in your research when you do. But what we'll do here is like show info is done and build H hydrogen bonds. So you can see here like serine of at the number of 397 is making a bond with the remdesivir. So there's one hydrogen bond. Okay, so this is the other information. Now what you do is write complex. Okay, so this is the first conformation. You got the energy, but you need a complex to further study the interactions. The other thing, you need to have this interaction. And also I have at the beginning I've shown you Chimera. You can go over there and save pictures as you want. So just go write complex and desktop, in the workshop folder, give any name. As I'm giving RAM complex. Okay, so it will be saved in PDB QD format, but you can change it later on by opening it any of the visualization software. So just save it. Okay, so okay, done. And then I'll show you. So I'm sharing the whole screen. So this is the Chimera, and you just go this one workshop, and I think here the complex. You open it. Okay, because it's a PDB QT format, so it is asking like which format do you need. So PDB QT is not there, but we'll set PDB file. So here is your proteins. So I think because I overlapped both, already there was a ligand and I overlapped this one. There's a problem with this one, I think. So you will get results like this. So select residue structure. Okay, like it. So I think So this red one is your ligand. I think if you get some disformalities in the ligand as we are getting, so I think we need to minimize first the ligand. If any problem comes, you just go to the ligand one and just make it perfect. And also with the protein one, I think because I have chosen the raw one, raw protein, and just removed all the other chains. But I think once you should have a very good structure of the protein and the ligand for this. But just I'm giving you example, like this is what you get your structure. Okay. So I think this is fine. This is the final step of this document. This is all the information you get. And after this one, so basically docking, so just go, I'm just discussing a normal thing about docking is like nowadays why I've taken this 6M71 is RNA dependent RNA polymerase. It has very significant role during this COVID time. So we did this research on this one, and what we did is like we did a virtual screening. We took a lot of ligands, maybe in millions. So we did with the Autodoc Vina. That is a different tool. That is also of the Autodoc, but that is for the what do you call this? As for the virtual screening, and it automatically takes one ligand at a time and then goes to another. So it is like continuous process. It's not like this one. So we found like many ligand that was binding very fit to this 6M71. So there are a lot of research paper you can go and see at the virtual screening section or dockings. So if you can read, you'll get an idea like how to write about docking and what information you need from this. So basically, what we are getting here is a complex and the docking complex with the ligand and protein. The main part comes after this one, that is molecular dynamic simulation. So molecular dynamic simulation is just one second.
Okay, Dr. Puneet, so you are done with Autodoc?
Yeah, I think it's done. It's nothing more than this one. Just they need to process, and they might get some errors because of their system, and so they have to find out like that is not the Autodoc because Autodoc is fine. Just they have to find the path and other things. So if they will work it out, that will be done. That is not a problem. That will be sorted out.
Yeah, that's fine. I would also like to answer few of the questions from participant, about error. So
Okay.
I'm truly speaking and fairly speaking that even we are, including me and probably Dr. Puneet, is still fighting about error of docking and simulations. So this error is endless. And we can't say that if we are having 10 or 15-year-old experience of our 5-year-old experience on docking and simulation. So we always get error-free experiment. No, it is not possible. If there is experiment, then there must be some error, and error may be of different reason. Maybe after due to the path problem, maybe due to some of the file we chosen is not proper. Maybe suppose we choose the PDB file is not a good PDB file, maybe some structural issue. So maybe because of that, either we maybe do not minimize energy properly. So that may be one of the problem. And maybe we missed some of the step of Autodoc, not intentionally, but maybe sometimes it's happened because almost 20 to 30 step is there. So it's fine. Sometimes we missed it, right? Or maybe when we are making the grid box is not properly, is beyond the active side. So that may be one of the reason. So error is a different thing. And one question is from Abhishek Chahar. Probably he is interested in MDS, molecular dynamics simulation. So
Okay.
clearly, I'm speaking that this workshop do not include molecular dynamics simulations, but I request Dr. Puneet to just give an overview of how we can correlate docking, I mean, autodock or molecular
Okay.
docking with docking simulations. And then after we will take few of the questions like Indira Chakravarti is also
Yeah.
asking about Lamarckian and genetic algorithm, you
Yeah.
know,
Yeah. I'll come to that. Yeah.
so
So it's like so frankly, with my experience, is I'm sharing with you regarding the docking molecular dynamic simulations. So don't go blindly with anything. Just this is not like you got a protein from the PDB structure, just downloaded it, and just a ligand, and you did docking. In this docking also there is this formalities, but the the main thing is just don't push the buttons in the docking. Try to understand every step what it is, and if the error is coming, try to read all the comments in the internet. You'll get the answer, and in that process you learn a lot of things. So it's better to try to understand everything what you get in the autodoc. So that because every in research every problem is very unique. Your protein might be very unique, your ligand will be very unique. That is why that is how you get published. So just focus on that. The next thing is like MDS. Yes, there is a paper, like Gromax you can use, but that is only for the Linux one. Or so you can use, but it's a whole new, it's that is very, uh, as you can say, because it is in Linux and open source. So from the installation to running all the things, it's a whole new thing. If you want to learn, and it will take time sometimes. So and NBS. So because this is GUI in the docking, still you are getting very less like errors. Generally, it's very less. When you go to molecular dynamic simulation, in every step, you'll get some of the other things because if there's a minor any atoms going from here to there, so you won't be able to run MDS. So the answer is yes, the free software that is Chromax. You can download and install, and that can be okay. Regarding this, the Lamarckian or genetic. So these are the algorithms. Basically, what they do is uh, see if you're running any like these docking. So of course there is an algorithm behind this. So what it does, because it runs continuously, like for example I'm saying of the genetic algorithms, because so it like suppose you have a ligand, it is having a 10 bonds, okay, and atoms. So what it does, it rotates every atom with respect to the other 9 atoms. So in that case, you can see how many rotations is taking place in that single ligand. So what it does, it minimizes the energy. So it finds the best pose. Like if it rotates the first atom and then calculates the energy with respect to that atom, and then rotate the second atom and then it calculates. And this is a kind of algorithm that works. And finally they have a lot of conformations and a lot of energies, and they filter out the best one. So this is a kind of algorithms that takes place in this one. So reading about the algorithms and mathematics, and that is a whole new thing. So there's two things, like you want to use Autodoc and you want to get docking, and you want to develop the docking softwares, or you want to contribute. So there are two different things in that one. So regarding this one, the algorithms, you can read about that, and and you can choose whatever you want. So that's the thing. And so
So Chaitanya Chaudhary has asked a question. So can you please tell about some techniques and some softwares infotation?
Informat. Okay, okay,
Yeah,
okay. So, like bioinformatics is very vast. It's like what we are now just talking about is like structural bioinformatics of the structures, and there are genomics, there are other things. So you in which field you want to be, like here in this field in docking and simulation, I think you need to master molecular dynamic simulation, and also you need to have virtual screening. We did just one docking. You need to have learn algorithms that can run multiple docking at the same time, and that also with a minimum of computational energy. So that is thing. So I think molecular dynamic simulation, if you're going in this field. So
Okay, so I think we are done with discussion now, and we should stop here because it's an endless discussion of bioinformatics. Just I would like to add to the last question of Chaitanya that which software one should master in bioinformatics. So my answer is whatever software you need for your project, for your experiment, or for your research. I think that software you must to be a master that software. So it's not like that. Suppose I am using Autodoc for my experiment, so I say that yes, you should master of Autodoc. But maybe Dr. Puneet is using simulation, so he will tell you that no, I think simulation you must be master. So it's not like that. It
yeah,
depends
yeah.
on your experiment, your
Mhm.
project, and maybe your research idea. And even it is not necessary. Suppose someone is knowing simulation and someone is not knowing simulation, and only he or she know only docking. So is it not like that this one is big software and this one is small software? Is not like that. Is all depends on your need. So that's all, I think.
Yeah, yeah. True.
Okay, so now the last thing to this session is remaining is a photo session with the participant and our expert, Dr. Puneet. And before we are going for photo shoot, I would like to convey my sincere thanks to Dr. Puneet for such a wonderful session. And I would like to frankly add this, that it is not possible even no much easier than this session on Autodoc. So whatever easiest way of training on Autodoc possible, Dr. Puneet did today. So thank you, Dr. Puneet, for this. And I would like to convey my thanks for accepting invitation for this technical session in spite of your busy schedule. I know that you moved recently to South Korea
Yeah,
and
it's
you
okay.
are
Always,
currently in the
always, no problem.
quarantine time. Uh, Probably tomorrow you will end with your quarantine.
Yeah, yeah, yeah. Tomorrow is
So
10th day. Yeah.
we also wish you good luck for your new research domain and
Oh,
new
thank
lab.
you so
So,
much.
on behalf of organizing team, and
Thank you
thank
so
you
much.
so much for such a wonderful session. And I also want to convey my thanks to participant for interacting with speakers. And I promise you that we will in touch with you. And whenever you need any support related to bioinformatics, we always happy to extend our hand. So me and Dr. Puneet usually always ready to help for particularly in bioinformatics. So you may contact us for this talking things. And I hope you must learn this Autodoc today. I'm not saying that you became master today, but yes, it must be a beginning, right? A good beginning. So thank you,
Sir,
Dr.
there
Puneet, once
is
again.
another question. Which software used to identify protein-protein docking interface?
Yeah, it was
Shafiqul
Chimera.
Rahman.
Yeah, that is I'm writing this.
Okay. I request all the participants to please turn on their cameras. You have already been provided with the panelist rights. So I request all the participants please turn on your cameras so that we can just memorize this moment forever. Please turn on your cameras.
They are allowed to open their camera, Dr. Mansita, by
Yes, sir. They have been provided with the rights.
okay.
So I would request all of you to please turn on your cameras. So many of the participants are actually denying the rights for the panelist. So we had already requested you all in beforehand to please keep your cameras ready as and when we might ask you. Okay, I request my student volunteer Akshita, please take the screenshots.
Yes, ma'am, I've already taken.
Okay. And we have also posted some of the screenshots in the group. Thank you so much to all the participants for being a very attentive crowd today, and thank you for asking all the questions. Tomorrow's session, we have already posted the schedule to you, so please join accordingly. In the schedule itself, you have got the link for the session will remain the same, only the link for valedictory changes. So that will be posted to you in due course of time. So please be there tomorrow on time. And if still someone has some questions, our esteemed guest technical speakers, Puneet sir is still here, you can still ask the questions.
Yeah, you can ask anything. So maybe also I'll be available later on as well. And Dr. Ravi, will be there always to help you, no problem.
Yes, and those who would be wanting the recordings of the session with the due permission of our speakers, we'll be sharing the same with you all. Okay, so thank you. I guess we have taken the screenshot.
Thank you, Dr. Manishita. So nice.
Thank you, sir. Thank you.